imagine that you are looking at a supercomputer which hardly needs any energy, which only works on 160,000 components. That is what a fly brain is. Now, you can just look from the outside at that supercomputer and try to guess how that works. Extremely difficult, right? So what you actually want to do is you want to go into that supercomputer and look at how these different components are wired up. And that's what the Connectome is. This required the collaboration with many different institutes around the world, many different labs, and even companies like Google, which worked with us to do, for example, a lot of the AI predictions.
Our lab joined the FlyWire community effort, where the first brain was being mapped. And we started mapping the gustatory part, the sense of taste. The technology we use, the modality we use to visualize the brain is electron microscopy images. Basically, they slice the brain of a female fly, and using computer technology, these sections are registered into a 3D volume. And then we use deep learning AI to basically trace each of the neurons and their structures and the connections to each other. And we also know that the sign of the interaction, so if they are activating or silencing the next neuron. And we do it brain-wide. So it gives us an entire map of the brain. A good analogy would be maybe the genomes, right? The entire sequence of the human DNA. Now we have the entire connection set of a female brain and now recently a male brain.
And so now we have access to what the supercomputer does, but we also have access of how it looks inside. So now we can start really decomposing it, analyzing it much more efficient, because we can now make really strong predictions of what these connections do and now test them by breaking them up, creating new connections.
There's a couple of things we took advantage as we were helping contribute to the establishment of this Connectome. One of them is to actually identify complete circuits, complete networks that goes from detection of visual signals, in our case, this is what my lab is interested in, into transforming them into actions. And Mert in my lab, at the time when he was a graduate student, took advantage of this data set to ask the question about particular pathways in the visual system that are very important for providing information about how the animal moves through the world. And so using this Connectome, he was able to identify neurons that we consider were really important for this information. He then developed transgenic flies that were getting us access, direct access to record and understand the activity of these neurons that we had identified previously in the Connectome, but also to manipulate their activity and evaluate the consequence of the activity of these neurons on the behavior of the animal. And the one really beautiful thing that came out from this work is that we have identified a structure of a network that we see in many other animal systems and in many different regions of the brains of these animal systems, including the mammalian brains like ours, that suggests that similar functions that we have ascribed in this fly likely are also present in these other regions of these other brains. And so I think that the power of using the Connectome data is the possibility to link what those structural configurations relate to computations, provide a sort of hypothesis generation that one can then test in physiology, and then compare the structure and function, and then actually really understand canonical computations that exist in many different brains.